So for the next part, we are going to discuss the challenges that we have with pandemic. So these are the questions that we have. I'm sure that you have heard that we put together a guideline. We will start from India. We have Dr. Venkatasubramanian from National Institute of Mental Health and Neurosciences, Peyman and Nastaran from Iranian National Brain Mapping Laboratory. Okay, now we go from Iran to Italy. And we have two brilliant colleagues from Italy, Mauro and Giovanni. Now we have Dr. Tony George from Canada. Now the next speaker, again from Canada, Dr. Laurie Zawertailo.
The remotely supervised protocol that we had just happened to have developed that's become very timely for its use.
And what I wanted to do is actually not speak to you for that long, but to just tell you a little about what we're doing at Wake Forest.
Okay, good. Now we can go ahead for the next part of the webinar today. We discussed about really kind of great hopes for the future, but Dr. Walton, now we are going to talk about the negative side or the challenges that we have right now with running trials with the pandemic and discussing about how we can deal with complexities that we are already facing. I know that all labs across the world are facing these problems. So let me kind of share my screen and show you the agenda and the list of talks that we are going to have. So for the next part, we are going to discuss the challenges that we have with pandemic and how we might be able to help each other to find solutions and move forward with the complexities that we are facing with the pandemic and the kind of the potential solutions that we're going to have. So today, Colleen, Dr. Hanlon, and me, we are going to I'm going to open the discussions. We will have some kind of talks from different countries across the world. And then we will have discussions with Colleen in terms of what are the potential solutions that we are going to have. And we also discuss about the guidelines that we have recently published in a paper in Brain Stimulation Journal. That might be something helpful. And I'm hopeful that kind of Leigh would be discussing about that. I'm not sure if Leigh is online with us today or not, but we will discuss about those things today as well. Okay, Colleen, do you have anything for the introduction or should we move forward?
I think we should just go right into it.
Okay, good. So in terms of the challenges that we have for tES, TMS labs around the world and the questions that we are facing right now, we have challenges in terms of how to move forward for remote screening, online assessments, and follow-ups, and what are the protocols that we can take into account for these remote screening and online assessments, and what are the challenges that we have. For example, here we use NIH Toolbox for neuropsych assessments, and we already have challenges in terms of, okay, if we do all the assessments online, can we do neuropsych assessment online, and what are the modules that could not be assessed online, and we need to kind of do them in person, and what are the alternatives for specific tools that we have to use in-person interactions to be able to do them, or there are alternatives that we can change them to other assessments. So there are challenges for that, and we definitely would be happy to discuss about that. So I'm going to just kind of share the questions and challenges that people are facing and some of the experience that we have here at Laureate Institute for Brain Research and the experiences that we are doing right now. And then the other question is, do we need to do COVID testing? And that could be an interesting question for people across the world, those who already kind of work in different hospitals. Do you do any testing for your subjects that you are recruiting for studies? Have you started to think about kind of doing any sort of testing for subjects? And you know that is something like a dynamic process. I was talking with one of my friends kind of a few days ago. It seems that right now, many international airlines, if you want to get to the flight for international airlines, you need to get tested before. So you need to provide a testing document that you have been tested something like 3 days before to be able to get into the flight. So is there any kind of plan to do testing before putting a subject inside an MR scanner, or have people started to think about potentials for that. For example, in our institute, at Laureate Institute for Brain Research in Tulsa, Oklahoma, what we are doing right now, we are using a device. It's called, I'm sure you have probably heard about that, Sophia or Quido, and it makes rapid testing for surface antigens with using a nasal swab. And it takes something like 15 minutes to do the testing. And that is what we basically started to use when we have been recruiting subjects. And last week we had challenges in terms of finding the kits for being able to do the test, and we basically had to start to change our protocol in terms of recruiting subjects without doing the testing. And a few days ago, we have been fortunate to be able to find a few other batches of testing kits. Now we are back to the original protocol that we had to be able to test every single subject that we are recruiting. But it would be nice if we have feedbacks from colleagues across the world. What are the strategies that are taken into account in different countries, and how they are thinking about doing testing or not, or how to move forward with that. That could be an interesting topic for discussion. And then, do we need to consider special considerations for different clinical population? As an example, I already have an ongoing trial that we are recruiting people from residential centers. And as you can imagine, when we bring a patient from a residential center and we do testing, and then we escort them back to the residential center, one of the challenges and considerations or concerns that we have is, okay, they are living in a closed community, and we bring somebody out of that community and do some kind of assessment, and then we put them back. Should we consider them as something like high-risk population because they go back to a community that is pretty much a closed community? What are the special considerations compared to, let's say, recruiting healthy control subjects from the community? So do we put them in extra risk? And that is something that we are discussing right now with the IRB and some other colleagues here in Oklahoma. And then I know that there are kind of challenges right now for imaging centers. So I've heard from different colleagues around the world that they are back to their own trials, but they cannot still do the imaging part because the IRB was not approving doing the imaging part. And even those who are back to do fMRI again, they have challenges in terms of, okay, how to change the room circulation, even in terms of the air conditioning and kind of air circulation, in a way to be able to get sure that it's not going to kind of make problem for subjects, because obviously you know that we cannot use masks in the MR. It could be really complex if you want to use masks within the kind of the MR coil, especially when you have a small coil and we have several other things. For example, right now we are doing a concurrent TES fMRI, and we already have something like 10 channels of electrodes over the head and the kind of the really narrow coil, and how, can we use the things like mask or not? So those are the challenges that we are facing right now. We are really interested to see what are the experiences that people have across the board. So we will have some speakers that would kind of present their experiences, but we definitely the floor would be open for those who have other experiences around the world about this question and how they are dealing with these challenges. The other issue is about how the IRB protocol should be revised. Should we change the IRB protocol? Obviously, for the trials that we are changing the interventions. For example, we have a trial here that we are doing cognitive rehabilitation for drug users. Now we have changed that to an online kind of intervention, and for that we had to basically change the IRB protocol. Or for some other trials, as we had two parts, a screening protocol and then recruitment and a kind of assessment protocol. As we changed the recruitment, because we had to do the testing, so we changed the recruitment protocol, but we didn't change the entire kind of the intervention, because we didn't change anything in the, let's say, brain stimulation imaging part of the protocol. So we didn't change that. But there are discussions, should we add a section on, let's say, extra risk for contacting COVID-19 during the trial? And at least right now, the conclusion among some of the colleagues that we are working together is we are not adding extra risk compared to, normal life that people have in the community to them. But there are definitely other considerations. Should we add anything to the IRB protocol or the consent forms regarding contacting COVID-19 during the trial or not? And the other issue that I think every single lab that is doing the brain stimulation or any sort of intervention right now with the COVID-19 and the pause that we had is how we are going to do data analysis when we have recruited something like half of the subjects, and then we are going to recruit half of the subjects in a totally, or at least some kind of modified version of the trial that we have been doing. For example, right now people are discussing about, let's say, some of the computerized tests that we are taking now with masks. And do we have any significant changes in terms of the mask? We still know that we are randomizing people between these interventions, but still that is something that should we report that or not? Should we consider that in the further analysis? And what are the reporting protocols for that? So these are the questions that we have. I'm sure that you have heard that we put together a guideline, something like 2 months ago. And the guideline is out right now. So you can see there are many details in the guideline. You can find the details. So we will discuss about the things that we have in the guideline with Colleen and Marom and other colleagues that have been involved in developing the guideline. So you can find many details about those discussions in the guideline. But we will also discuss about some updates right now that people are taking into account. Okay, so let's move forward with the speakers that we have today. We will start from India. We have Dr. Ganesan from National Institute of Mental Health and Neurosciences, and Venkat. The floor is yours, and we are ready for your talk.
Hello, thanks, Hamid, and it is a pleasure for me to be participating in this webinar, and I have learned quite a bit, and I look forward to learn more through the interaction. So our lab is primarily interested in doing transcranial direct current stimulation studies. We run this WISER neuromodulation program, which is the acronym that expands as weak intensity stimulation for enhancement and reintegration. So there have been ongoing clinical research studies examining several psychiatric disorders: schizophrenia, obsessive compulsive disorder, alcohol dependence syndrome, and neurocognitive disorders. We also do quite a bit of concurrent research methods, as Hamid was pointing out, in terms of MRI, EEG, and FNIRS. Can I have the next slide, please? So COVID has thrown open lots of challenges, and it has significantly affected our ongoing TDCS research studies. Like most other centers, all research studies were suspended in our hospital from the last week of March. And during this time, over the past months, we have been constantly working on measures to restart the research studies with careful evaluation of risk status. And in this context, the guidelines paper has been tremendously helpful. There are several SOPs that we have established. A couple of weeks before, our IRB has suggested that we can look at initiating these studies by following some of the procedures that they have recommended. With respect to the operating procedures that we have evolved to restart the studies, we have made questionnaires for remote screening for subject risk status, as well as we also have evolved a strict protocol that will continuously assess the risk of the research staff who will be closely interacting with these subjects. We have modified the consent form explaining the risk of COVID, though, as Hamid was pointing out, there is one issue whether we should take the risk more than what is there in the community versus not, is something which we are debating. And the online assessment has been incorporated in the modified consent form, which is being submitted to the IRB for review. And there are several operational measures that we have evolved, especially minimizing the time of participation through conducting video conference mode clinical assessment. Most of the clinical symptom scores can be reliably evaluated through a video conference mode, as well as the consent details can also be explained through the video conference mode. That is something which we are now piloting it out. the testing part, the COVID screen questionnaire that we have got is something which the hospital is using. So there is a risk evaluation, and once a person crosses the risk score where the antigen testing or any other COVID test is indicated, as of now we have decided not to include such subjects for research studies. And the requested protective measures to do brain imaging, it's a big challenge, whereas the FNRS procedures is relatively less challenging. So that is something which we are about to initiate. So what we have done very recently over the past week is we have done some pilot single session studies with healthy volunteers after a comprehensive risk screen to ensure that we are able to implement all the SOPs. And our plan is to initiate single session TDCS studies, which are primarily investigative research projects. And in that, to begin with healthy volunteers first, and then move on to patients. And depending upon how our experience unfolds over the next 2 to 3 weeks, we plan to initiate the intervention studies. So this is the kind of a roadmap we have thought of, and I look forward to interact and learn from the group. Thank you.
Thank you, Venkat. Thank you. Now we have Peyman and Nastaran from Iranian National Brain Mapping Laboratory. Peyman, are you online?
Yes. Hi. Hi, everyone. I'm Peyman Ghobadi, PhD candidate at Biomedical Engineering, Shahed University. I'm here today to share our experience with running TES fMRI trial during COVID-19 pandemic. First of all, I would like to appreciate Hamed for inviting me to give a lecture in this perfect webinar. My colleague Nastaran and I were conducting a TES fMRI trial to investigate the neurocognitive mechanism of TES in autistic people at the NBM. NBML, or National Brain Mapping Laboratory, provides facilities and data for the development and growth of brain mapping in Iran. Unfortunately, with the spread of coronavirus, the NBML was closed and our project was shut down. It has been closed totally from March 10th to April 11th. Please, the next slide. After the announcement, the government facility can restart their works with a less crowded environment. The NBML has started its work since April 11th with only 20% of its staff, and with the working hours reduced from 8 hours to 5. Due to COVID-19, we missed some of our subjects who were supposed to participate in our study. So in the first step, we announced through advertisement that we follow the safety protocol in our study, and the laboratory is set to recruit some new subject again. Please, next slide. We follow the guideline which Professor Marom Bikson and colleagues have published for conducting TES or TMS studies in COVID-19 pandemic. As you can see in the slide, all the laboratory personnel are fully equipped with the safety devices, and the equipment and furniture are disinfected very evening, and also eating, and in some cases drinking are fully forbidden here. Hand washing equipment, hand sanitizer, and temperature measurement for participants at the entrance are provided. Before participant entrance to MRI scanner room, the gantry and their radio frequency coil and MRI table are sanitized with cleaning products. In the brain stimulation room, in addition to masks, medical and research personnel are protective eyewear covering the entire front face. After the TES session is over, all the stimulation equipment, including EEG cap, cables, tape measure, electrodes, and sponge pockets are sanitized And finally, the stimulator trolley and treatment chair are wiped with an approved cleaning product. However, the MBML should provide plans for COVID-19 testing of participants and improve ventilation facilities for the stimulation and fMRI scanner rooms. I hope these details could give you a better sense of how we are trying to tackle with the novelties circumstance. Thank you very much for attention, and I hope we can get rid of or at least to adapt COVID-19 very fast. Thank you.
Okay, thank you, Peyman, for presenting the talk and sharing your experiences in Iran. Okay, now we go from Iran to Italy, and we have two brilliant colleagues from Italy, Mauro and Giovanni. So Mauro and Giovanni, I'm not sure which one you're going to present. We are here with you, and the floor is yours.
Thanks, Ahmed. I'm Giovanni Martinotti, and we are sharing with this presentation with Mauro, and we are speaking about the situation in Italy in our neuromodulation and addiction center. We reopened the recruitment in June after a period of stop, of course. Since then, 9 subjects have been enrolled after the onset of the pandemic. Actually, we treat 2 subjects per day to reduce, of course, the infection risk. All patients are screened for the COVID symptoms, and of course, we are doing the body temperature measurement before. At the moment, we are running 2 trials. One is about, and we are evaluating the BDNF, which is the brain-derived neurotrophic factor, before and after the treatment. And then we have another trial about gambling disorder. This is our systematic review that we have presented in the last few weeks about the results from tES and TMS studies. And in this systematic review, showed that stimulation instead of inhibition of prefrontal region could be beneficial to contrast dysfunctional gambling-related decision processes. And this was the base for our trial about gambling disorder that we are starting to run right now. Next slide, please. Okay, here you can see all the studies that we included in this systematic review. That is something that is useful for us in order to run the trial, which is the next slide, about the role of rTMS in gambling disorder. This is a double-blind design which will consider 50 gambling disorder patients, 25 active and 25 sham. It is a multicentric double-blind randomized trial with 3 centers involved, which is called the Break-Free Project. We are going to run an accelerated rTMS specific for gambling disorder. And apart from this trial that we are running, starting to run in these days, the other thing that I would like to mention at the end of this short presentation is the fact that we have we ended our double-blind study about coke and use disorder treated with rTMS. We recruited 80 subjects at the end of the trial. These results are very interesting, and we are submitting the paper right now. And let's hope to see this paper, and let's hope to have the possibility to present data about this probably in another occasion when we have more time to discuss about it. Thank you very much. I don't know if Mauro wants to add something, but this is what we are doing now.
Thank you, Giovanni. Mauro, do you want to add anything to that? Good. Now we have Dr. Tony George from Canada.
Thank you, Hamid, and thank you, Colleen, for organizing this session. So I tried to summarize everything about this in the title, so my apologies for the long title, but I'm a end user. I'm not a developer of TMS or TMS, and we have an interest in my lab in treating cannabis use disorder in people with schizophrenia, and also the mediating effects of cognition, given that's such an important domain in people with schizophrenia. So we're doing a randomized trial that's supported by NIDA. Actually, Dr. Walton is our program officer at NIDA, and it's been going on for a little over 2 years. If you go to the next slide, I'll sort of diagram to you what we're doing. Okay, so we call it the CAN-STIM study. It's an R21 supported by NIDA. And basically what our goal has been is to take a total of 40 people with schizophrenia and comorbid cannabis disorder and stimulate bilaterally the DLPFC, dorsolateral PFC, knowing that it's a key brain region that may be dysfunctional in people with schizophrenia, and also, you know, modulates brain reward structures. And we do standard high frequency RTMS at 20 hertz. We're fortunate at CAMH through our Temerty Brain Stimulation Center, which is a philanthropic center made possible by a generous donor, to have, you know, this area of expertise at CAMH. We also use the sham coils, so we use the mag-stim coils that does everything but generate the magnetic field for RTMS, and we randomize people to the active versus sham treatment for a total of 4 weeks of active treatment. We look at cognitive outcomes, we look at behavioral outcomes, including craving, cognition, and of course urine toxicology. The two young students down below, Carolina Kozak and Darby Lower, the grad students who run this project, and so who have been impacted by COVID-19. And one of the things that we do, I think that's modestly innovative to increase retention of these very difficult to recruit and treat subjects, is at each RTMS session that they attend, we give them progressive payments, usually as vouchers, to incentivize them to come for the RTMS treatment. And then we do a 1-month follow-up. So just in the last slide, you could move ahead there, Hamid. Oh, just, yeah, I'll just go to the, so there have been some obvious progress and challenges with this. So we've had to revise the number of people that we study. We've actually enrolled 24 people with those incentives. We've actually had 19 trial completers. However, since mid-March, like most of the clinical research at CAMH, we've been shut down entirely. We were coming towards the end of the first year of no-cost extension. So NIDA, through the efforts of Dr. Walton, gave us an additional year to finish this up. And you know what I would say is people with schizophrenia and cannabis use disorder are difficult to recruit, even though we have so many of them at our center. We have about 2,500 patients in active treatment. I'd say about 20 to 30% of them use cannabis, but you know, we've had trouble getting these people in before COVID-19 and obviously been shut down. So we're now planning to expand our recruitment to other hospitals that treat people with schizophrenia, and we're really the major center. And you know, obviously we're going through the same things. We're having to restart, get approval from the organization, from the IRB, and you know, another practical issue that's come up since we're an end user is that booking time for the RTMS procedures, which is not something we control, has turned out to be a challenge because they have limited staff available, and they've really had to use many of those research staff and clinical staff that support the studies exclusively for clinical care. So let me stop there. Those are just some of the challenges that we've had in the COVID-19 pandemic era in doing a RTMS trial in this very difficult clinical population.
Thank you, Dr. George. Thank you, Dr. George, for the nice presentation. Now the next speaker, again from Canada, Dr. Laurie Zawertailo. Laurie, are you with us right now?
Great.
Could you?
Thanks, Hamid. So yes, I'm at the same hospital as Tony, and we are doing a slightly different study, but probably have about the same types of challenges that Tony presented. So I'll just try and add to what he said. So if you just go to the next slide, please. So our study is in smokers, and it is a randomized clinical trial where everyone is getting 12 weeks of varenicline treatment, but they're randomized to either active or sham TDCS. And it's a standard 20 minutes of 2 milliamp stimulation over the left DLPFC. And then another kind of addition to this is that we're doing MRI scans prior to the start of treatment. So 2 scans before they start treatment, one under abstinent conditions and one where they're sated. Then they start the treatment. So on day 1, they get their first dose of varenicline and their first of 10 daily TDCS sessions. So the first 2 weeks, 10 sessions Monday to Friday. And then for the next 10 weeks, continue with varenicline and get bi-weekly TDCS booster sessions. So every 2 weeks. And then at the end of treatment, they get another scan, and then they get followed up at 6 months. So if you're interested in more details of the protocol, it is currently submitted and under review at trials. So hopefully will be published soon. These are the 2 students that are running this study. And this, again, is another challenge that we haven't really discussed, is how, you know, this affects timelines for completion of PhDs, which is a whole other level of anxiety for the students and the supervisors. So Helena Zhang and Noreen Ramani are running this study. Very anxious to get started again. So next slide. So we have, as Tony said, been shut down since mid-March. So it's coming up on 6 months. Complete shutdown. We haven't been able to do anything or really come on site. I'm on site today, but I don't normally come into the office, and neither did the students. So of course this meant that we had to stop all recruitment avenues. The Research Imaging Center is also completely shut down. At the time of shutdown, we had 2 participants that were currently in the study. So what happened was we were able to mail them their medication. So they continued with the varenicline part of the trial, but they couldn't do their booster sessions, and they had no end of treatment MRI scan. And they've both completed their 6-month follow-up. One was quit, which was great. The other kind of quit relapse, and at the time of 6 months had actually relapsed. We had 4 participants that were eligible and ready to start, and we've had to completely put them on hold. Hopefully they'll still be available and interested when we are able to restart. But this is, you know, as Tony said, these are really difficult studies to recruit for. Even, you know, even though we are looking for smokers, it's a really big time commitment. And so the fact that we have 4 people ready to start, and it has taken us 2 years, I think, to recruit the first 25, and we need another 25, you know, that was a huge blow to us. So next slide. So we have had some communication from the powers that be at CAMH that we are restarting. So clinical research is restarting first, and because this is a clinical trial, we were able to submit for approval to restart. So we're at the kind of first phase, which is kind of higher up approval by the chief of addictions, and now we have to get approval from the research services office, and if we have any amendments to our protocol, those need to be approved by our research ethics board. So we don't really, the only thing that we're changing is that our telephone screening is going to be done off-site at home where possible, so that we have fewer people coming in. We are only allowed to be at 25% capacity, both in the clinic and in any of our research. So, you know, only 1 student probably at a time, and maybe 1 clinical staff. We still have not had, well, some indication that the imaging center is moving towards reopening as well. So we're hoping that by late September or October, we'll be able to at least start recruiting again. Thankfully, all of our assessment tools, et cetera, are already on REDCap, so we can do them kind of over the phone or remotely if we need to. And then, we're just trying to figure out everything or get direction on all the sort of disinfecting protocols. We know that the research staff is going to need to be in full PPE. We can't maintain a 2-meter distance from our participants when we're applying the stimulation, et cetera. So we need to figure all of that out just to keep everyone safe. We do have a standard COVID screening protocol and process at CAMH, so they will have to undergo that every single time they come in.
Thank you very much, Laurie, for the nice presentation. And obviously, we all have the same challenges across the board, and we are all together in these challenges. So hopefully, we can find kind of collective solutions for these challenges that we are facing right now. Okay, the next speaker is going to be Dr. Charvet, Leigh Charvet from NYU. Leigh, are you with us right now? Hello, Leigh.
I'm here.
Hello.
Yeah, hi, I'm here.
Good, good to have you with us. Do you have any slides?
Yeah, so I don't know if with Dr. Hanlon, if we want to do a discussion, or I can just kind of run through some slides. I'm here at NYU in New York City. We're, COVID hit us early and very hard. And so my background, I'm in the Department of Neurology, and I work primarily with TDCS, providing TDCS for rehabilitation and also for treatment of depression in people mostly with neurologic disorders. And we, for a variety of reasons, I came from a background in telerehabilitation, and so we've spent a good 6 years or so developing what we call remotely supervised TDCS or RS-TDCS. And this is where we have a very rigorous process using live video conference to provide TDCS to patients at home. And because we work with people living with neurologic disorders, we spent tremendous effort to have the procedure really applicable to almost anybody. So we include people who are very severely motor impaired in some cases, for instance, wheelchair dependent with limited use of their hands, or have somewhat severe cognitive impairment. We've gone up to full age range from 18 to 80 years old. And so we're really confident in that this is a way for home-based delivery of TDCS. And so when COVID happened, we were in position that we were already doing at-home trials. And so we, from that point, only had to change our baseline and study in visits to go remote as well. And we've been able to continue during this time. So we've actually enrolled, I think it's been 53 people since the COVID pause, and delivered almost 1,000 at-home TDCS sessions. And also to note within that, we have a clinical service as well, where through something called Innovative Care, we provide home-based TDCS for patients, and that's been able to continue also during this time. So that's kind of the broad background, the perspective that I can bring, and just a vote for home base. And so, Dr. Hanlon, I don't know if you have specific questions, or I did want to orient everybody to the paper that was shown before too, that was in brain stimulation. It's a very nice structure of response and then recommendations to getting labs and personnel ready. So we can talk about that as well too.
Yeah, do you have any slides for that? Do you want to just discuss it, just navigate through the paper yourself?
So if that's coming through, so that's just a snapshot of the paper that's a nice orientation to response, and also a very nice survey of how people have responded to the COVID challenges during this time. So there's a 3-phase model that's proposed in the paper with the immediate response. So for instance, all of us had to respond immediately when we were presented with pretty much a sudden shutdown. And I think that was pretty much across countries in that we had to act very quickly. And I think now we'll all be prepared for something. but that was a sudden shutdown that we certainly experienced, where we were told no more enrollment, and all lab personnel had to immediately not come to work. And so phase 2 phase 1 is starting to respond from that, in characterization of some of the strategic responses with all these evolving restrictions. We've had a lot of stop-start kind of messages in our institution, and also trying to respond as well to the varying guidelines that mentioned here as far as terms of testing and state of PPE and sanitization and all kinds of challenges. And then phase 2 talks about definitely being prepared for future events that hopefully that we won't face again, but I think we'll all be in a better position, having learned the hard way from going through this. And it talks about optimizing productivity and going forward from there. And so I just wanted to again highlight to the audience that the paper includes some very nice set of guidelines and kind of talking points about how to be in preparation. I won't read them through here, but initially how you can be in position to move forward. And then phase 1 talks about, for instance, converting trials, or maybe even having on hand a provision to go remote, should you need to be, when you get your initial approval for the trial. And also importantly, having lab personnel be able to work from home. We were able to use lab dedicated computers where our lab personnel deliver the TDCS treatments, and so that they're HIPAA compliant. Everybody had their own computers that they work from home, but also the lab provided everybody a dedicated computer where they could safely connect and compliantly connect to our patients through video visits. And then the next phase is talking about the future, and again, being in preparation. And then also, I know there's a lot of subtleties to work through, and also I think it's very powerful that was mentioned about the impact on timing for graduate students, and thinking through that. There's just a lot that I think we have to consider, and then extensions for our grants and our study timelines of, especially for when we're required to be on site. And then this is just a snapshot of, I'll move through, remotely supervised protocol that we had just happened to have developed, that's become very timely for its use. And basically, so we give patients dedicated computers, and we've worked out, we control the computers remotely. We use remotely controlled TDCS devices. So we really take on all technology burden on our side. And we have a very, our initial goal was to make it as rigorous as on-site brain stimulation. So we have a very rigorous stop, no-go kind of criteria that's applied at every session. And every session we're currently live with the patients. And I also just like to make the point along the way that I think, recruitment for on-site studies is very difficult. It's a challenge we all face, especially when we need repeated treatments, which is definitely the case for TDCS. And so telemedicine just really meets many more needs than just convenience. It's, there's geographic requirements for what's considered telemedicine, for instance, for Medicare. But here in New York City, most of our patients are within 5 geographic miles of our clinic, and still telemedicine, it fulfills a tremendous need. So we actually captured just how much time patients spend in terms of coming to clinic, and the cost just in commuting. And it really adds up. If you, for instance, did a grant budget just for each patient's cost in terms of time and also transportation costs just to come to daily treatments, you can really make a compelling case why at-home brain stimulation for study as well as for treatment is really helpful. So just to review the key components we use, so TEREX is mini-CT, which is a pre-programmed device. It can do active or sham current. For double-blinded trials, it's really optimized for easy patient operation and includes abort keys. And we went through many iterations of a headset that patients who maybe even had limited use of their hands could place while at home. And so these are cap-like headsets that we use. And the electrodes snap right on. So they come in single-use packets and snap right on to the headset and then are disposed after. And again, as I said, we connect in real time and we take on the technical burden ourselves. So we actually have terminals where we call our lab personnel RS-TDCS technicians. So we can deliver approximately 20 sessions a day to patients, both in clinical service or in research trials. So it really is scalable in a marked way. And it's as tolerable as you would expect. Really, we found absolutely no difference from on-site delivery versus at-home delivery. So just the profile that you would expect. One feature is, though, that patients are very compliant with treatment. We have a strong demand for these treatments, and our patients will complete, we've had trials where we go up to 60 days of treatment across 12 weeks, and our patients, we have across studies about a 96% protocol completion rate, and we also have very rapid recruitment. And so this is definitely meeting demand and interest for patients, and I think for non-drug treatments that they can access from home, and there's just so much interest in brain stimulation therapies. And so again, just as I mentioned in the beginning, in our response to the COVID pause, it started suddenly for us in March, and we were able just to convert those baseline and treatment and visits to on-site remote processes. We use RedCap. We have had to increase our shipping and receiving division of our lab, and that we have shipped equipment now. And so that's a little bit of the complexity, and safe, reliable shipping, cleaning it. We have incoming and outgoing equipment, cleaning protocols with the system marking what's been sanitized and what hasn't, and tracking shipping and that kind of thing. And also we have a protocol now for our first visit, the baseline visit, where we help the patient unbox the equipment and orient to everything. And we had 3 ongoing clinical trials at the time, targeting 30 sessions or 20 sessions, one with cognitive training and one with motor training hand exercises. Both have continued enrollment. The motor training required a little bit more finessing in terms of the equipment that we had to ship, because not only are they getting the TDCS equipment, but they're getting the motor training equipment. And then we were in the same boat with as was described before, with our imaging trial, where we have a simultaneous MRI TDCS study underway, and we've been able to resume, as of 2 weeks ago, we've been able to resume on-site MRI. And then also with our clinical service, so there's a lot of interest and at-home TDCS for clinical service. This was largely developed to try to meet the needs. We've been kind of overwhelmed with patients who want to try TDCS based on what's been published in the literature, or we have people coming from our trials who want to extend treatment. And so we have a small option for clinical TDCS at home. And this has been able to, here we use our video visits through Epic, just as we would with our clinical visits. And so it's run as a clinical visit, but using the TDCS, the RS-TDCS procedures. So that was kind of a rapid overview of our perspective and just a vote for at-home use. And I think I'm excited when I think a lot of the devices and the advances are coming through devices, and I think that they're becoming more portable and more wearable. And I think that we can easily use newer developed devices in the context of this protocol.
Thank you, Leigh. Thank you, Leigh, for the great presentation. Just a kind of a simple question in terms of IRB application. Have you had to apply for an IDE for the kind of home-based devices, or what was the process for that?
Yeah, no, these are cleared devices that Soterix has. And so we have not had to apply for anything specific for our protocol. We use the device as it is within the context of our protocol. So we've not had to apply for anything.
Good. Thank you very much, Leigh. Colleen, now kind of the floor is yours for the discussions, and we definitely have colleagues from different countries across the world. So that would be nice if they also have time to discuss. We will have 20 minutes for further discussions and sharing the ideas and questions that we have from participants.
Great. That's awesome. Leigh, thank you so much for giving that presentation. I think the remote TDCS is fantastic. I think all of us probably would love to be able to be giving treatments and interventions remotely right now. So I hope that you can kind of apply some of what you've already done to addiction. That'd be awesome. So we have about 15 minutes left. And what I wanted to do is actually not speak to you for that long, but to just tell you a little about what we're doing at Wake Forest, and then really leave as much time as possible available for everyone to kind of have more of a discussion about unique things that are happening at your institutions. So at Wake Forest, we've been pretty fortunate. The COVID burden here has not really been very high compared to some other places in the world and in the United States. And so I'm very thankful that we were allowed to open our studies in sort of June and July. But it did take a lot of preparation and planning. We currently have 4 studies that are ongoing and recruiting in the addiction space. These are all clinical trials. So when we kind of in May and April, when we were preparing, we were watching the numbers, and alcoholism rates or alcohol consumption is rising during COVID. The stress and isolation associated with COVID is causing people to smoke at higher and accelerated rates. And so, you know, you look at those numbers and you think, my goodness, now more than ever, we really need to be helping people. And so we sort of made that plea to our IRB. And we were able to reopen our alcohol studies and our pain studies first. We actually are just now starting to get permission to bring our smoking studies back. And I'll get to that in a second. Hamed set up initially when he kind of created an organization for this center, for this session. We now provide individuals the option for a remote screening and consent visit. A template was created by our university to create a pathway for many people to do this. We don't require people do it remotely. And honestly, thus far in our studies, everybody is coming in person. I don't think we've done any remote consents, honestly. Mm-hmm. So people are choosing to come in. We also, for every visit, we have a COVID screening checklist, which a few other people have brought up. This was also provided by our institution. And you can find a lot of these online. Actually, this one I did just pull off the internet shamelessly from University of Massachusetts at Amherst. But it's very similar to the one that we use. We also take everybody's temperature via one of those little forehead thermometer checks. And you have to be under 100 degrees in order to come in. So we do this every day for every person. In terms of special considerations, as I mentioned, we were able to open our alcohol study and our chronic pain studies first. The IRB was a little bit more concerned about our smokers, because initially there was some concern that they would be at higher risk for having particularly bad outcomes associated with COVID. So while bringing some of our people in, like people in chronic pain, was still seen as a higher risk to benefit ratio. For smokers, it was unknown. So they recently lifted that restriction, and we will be able to bring those people in, which we're excited about doing. We advertised for all of these studies, we advertised for our participants in the community. We use the newspaper and the internet and varying other avenues that we have. We actually find plenty of interest. So that was one of our concerns, is that the population really would be a little bit concerned about coming into the medical center, which would be rightly so. But in fact, that hasn't seemed to be too much of a burden. We still have a decent amount of interest. We've noticed that, especially in our alcohol users, most people that have their treatment-seeking alcohol users, most of them that come in say that while they were at home with COVID and they were supposed to be taking care of their kids and their spouse was home, they became more and more aware of their risky drinking behavior. And so something about being at home and being surrounded by these other stimuli really pointed out that they had a problem that maybe they should seek help care for. So we're really happy to see that, right? We feel like we're hopefully making a difference in the community. There we go. Special considerations for special procedures. We do have our MRI center also opened in July. The volume is really low. I mean, the number of scans, there has to be a half an hour between each person in order for the techs to clean the scanner. But all participants are required to wear a mask in the scanner. So initially we thought that might be a challenge. I think we've maybe done 15 MRI scans since this has started. And we've only had one person go in and have the mask on and feel claustrophobic. So, which actually really surprises me. I went in because we wanted to sort of pilot it, and I went in and I'm not claustrophobic, but it is a little different. So I've actually been oddly surprised that the participants seem to be coping with it pretty well. We also, especially for our alcohol study, for breath alcohol, we typically have a common device with disposable tips that people blow into. And so, so we felt like that probably was not safe to do because there's a common filter, right? This kind of thing is actually talked about in that review article that Leigh and Hamid have brought up and a few other speakers. But thankfully, the company that we get our BAC meter from actually has these disposable keychain-based BAC meters. So we bought a few and we did some kind of quality control testing to see if the precision was comparable. And although it wasn't exactly on spot every time, it was pretty close. And so, so we actually changed our protocol and we just bought a ton of these disposable keychain breathalyzers, which actually, so we give them to the participants and they take them home. And they really, they really like them. It's kind of like an added perk of the study. So that's actually worked out really well. I think that when COVID resolves, I think we'll probably stick with the keychain-based BAC meters. They're really pretty great, and they're not that expensive. And then so for our smoking study, again, that was delayed a little longer, and we couldn't find an equivalent for our CO monitor. There wasn't really a disposable CO monitor available. If anybody out there knows where to get nickel or nicotine testing strips, I would love to know, because we also have found it really, really hard to find them and purchase them. So we've had a little bit of challenges in that regard. This Smokalizer is who produces our CO monitor, and they do have sort of a COVID statement about the filtration of their device, and we've had some communication with them, and we've sent it to our IRB, and we're still sort of going through the approval process there. So that's a little up in the air, not solved yet. If anyone has good solutions, I'd love to know. And then finally, most important in all of this, right, is sort of the health and wellness of you and your team. And so we wear a lot of PPE. Our lab goes through $100 in PPE every month right now. So that's a lot. Giving TMS, we wear face shields when we're actually up against the person. We have a standard protocol for washing your hands when you come in and out of the room. So we make sure that essentially there's a seal, an invisible seal in front of the room. And so you wash your hands when you go in, when you come out. Face shields on the people delivering the TMS. We are not required to wear gowns, but some of the people on our team would prefer to do that. And so I, as a lab leader, I'm happy to provide them with all of the PPE that makes them comfortable. And I would suggest that you all do the same. And in general, I actually think in our case, I think the team camaraderie is fantastic right now. There's this group think of like, we're in it together, we're working forward and towards something. We have people every now and then that sort of, they travel somewhere else, so they won't come back until they've passed for 2 weeks before they'll come back into the lab. But we have enough overlap that we'll either pause their study or pause their participants, or we have people to fill in the gaps. So yeah, so it's actually working pretty well for us. And that's all from here. So maybe, Hamed, we should transition to asking people if they have any questions.
Yes, of course. Yeah, I mean, whatever you decide in terms of the discussion and how people are interested to share their ideas. I mean, we are going to kind of chair the discussion panel. So go ahead.
Sure. Well, I guess in the same way we did before, does anybody feel free to raise your hand if you have a question you'd like to ask or a story about what's happening at your site you'd like to share? Yes, Eduardo.
Yes, thanks for a great talk. I just wanted to ask about, so we haven't started yet our trials, but luckily I finished one right before everything started, so we haven't had that problem yet. But I was wondering about the screening procedures. What do you think has been better? Because the clinical screening, I think, for me as an MD, I think is better than the Sophia 2, but I don't know the experience with the Sophia 2 that Hamed mentioned, because there's a lot of false positives with that, and it's not even used as a, for the diagnosis, and PCR is way too expensive. So I was wondering what you think about that. What would you prefer? Because we will have to implement it here as well. And I will lean towards the clinical one in my case, for example. I don't know what you think.
When you say clinical, you mean like questionnaires and temperature check?
Yes.
Yeah. In our case, I'm happy to say that burden has been taken off of our shoulders, because that's what our university requires. And so that's why we do it. But I'm certainly open to, Hamed, why maybe you can provide some insight.
Yeah, that is something that I think that it's basically the decision that we are not making. Most of the time, this decision that is made by the institution. So I know that people in different institutions, they have different kind of decision-making process in terms of how to and depending on where you are located, whether you are inside the hospital, and even in terms of the availability of these kits within US, if you are in different states, you have different access level to these things. So there are states that you cannot basically buy these kits, and there are states that you can buy. So it depends on many of those really not very much scientific, but mainly political or kind of organizational decisions that people need to make. But it would be nice to have a kind of idea about the variety of decisions that different institutions are making right now. So it would be nice if others have different experiences to share and see what are the variety of different kind of potentials that people are exploring right now around the world. Betty Jo, do you have any?
Yeah, we're purchasing one of the Abbott machines and are going to be testing regularly, because I'm with 40 to 45% of cases being asymptomatic, I'm really not comfortable with using a symptom screen as your primary method of ensuring that you're not bringing people in who could be infectious. So we are going to be, we're doing symptoms and temps, of course, also, but we are going to be testing regularly everybody, staff and participants for the COVID. And I guess in terms of other things people were talking about, I think we do need to mention the risk of COVID especially for the TMS procedures, because it is a little bit higher than the community risk, because you do have to be within that 6 feet radius for a prolonged period of time with somebody that you otherwise would not come in contact with. So that does pose some sort of risk, and at least in our situation, we're mitigating that, I think, pretty extensively with the frequent testing. But participants are owed that information so that can help them make their decisions. I have a little concern about the PPE issues for when you're actually delivering the TMS. We've been told we do have to have a mask on the participant, which seems to me that anything that is escaping is being directed right towards the person who's administering it. And yeah, with these face shields generally kind of angle out a bit at the bottom, and I wonder how much protection that really does provide. So I'm kind of interested right now. They don't want us to use up any N95s, so they're not letting us use the N95s. We will have our participants regularly tested, so I think that helps a lot. But they're telling us we'll be using surgical level 2 surgical masks and face shields.
Yeah, we thought about the so our participants all wear masks, of course, not N95 masks, the same sort of surgical ear loop masks. But we thought about whether or not we should have face masks on our participants. It's not required by our institution. Again, I think a lot of this you have to consider as well the sort of community transmission rates in your area. As an alternative, we thought about having glasses or goggles. So one of the keys, so your nose and your mouth are covered by your mask, right? But you can put your fingers in your eyes and things. And so an alternative would be to have them wear some like goggles to cover their eyes, which then wouldn't kind of affect the sort of air circulation towards the person delivering the TMS. So that's one thing that you might want to consider.
Also, for anybody, you guys have been delivering the TMS already? Because I'm a little uncomfortable too with having the participants' face pretty much entirely covered and being able to see their response to what we're delivering. Is that, have you been comfortable with what you're seeing in terms of being able to pick up the twitches and the jaw clenches and things?
Yeah, I mean, hopefully they're not twitching too much, or we're probably doing something wrong. But we talk to them throughout the treatment. So as you know, in our case, we're doing ITBS or CTBS, so it's pretty quick. And when we do the ramp up, we ask them how they're doing, and so they're responsive to us. I mean, it's certainly a good point that we see less of the participant when they're behind the mask.
Other comments from other colleagues around the world in terms of the solutions and the challenges that they have. We already have some comments in terms of the cotinine levels and the other things in the chat space, so colleague probably can address them. But if there are some other questions and things that people are interested to share.
So I see that Christine Sheffer posted something about a take-home CO monitor. Thanks so much, Christine. That'll be great. If anybody else on the call is interested in that, she posted that in the chat window. Is anyone else actively running TMS or TDCS at their site?
This is Chris Sheffer. Yes, we're finally up and running at a good volume. We were shut down. I'm in New York. We got hit pretty hard, and we were shut down for 3 months. And we just worked with it seemed impossible at first to get up and running. Started in June, took us probably a good month to get all our procedure working with infection control. And they don't solve the problem for you. You have to kind of work with them to kind of solve the problem. But my coordinator, Amy Lynn, and the team, it all seemed really awkward and weird and tedious at first. But now Amy Lynn, I think she's on here, she might say something different, but I think it's much more routine. I'm very glad to share our, our protocol, our safety protocol with anybody who wants to look at it. It was approved by our infection control. I'm at a cancer center, so, we are always really concerned about infection control, but, we have the full PPE going on, temperature, self-reported symptoms, tracking that self-report in case anybody does come up positive at some point so that we're able to track. Recruitment was really a lot of effort and in a different way than we usually do. But, the people the people who are talking to us, few of them say that it's an issue or want to know about, but I think the people who it's an issue for just don't call us, if they're worried about exposure. I don't know, Amy Lynn, did you want to say anything about how difficult it was to kind of put in all these new procedures? We can't see as many people in a day, right? Because we got to we did an hour between sessions in a room, and now I think we're down to 30 minutes of cleaning.
Yeah, we haven't had too much trouble once we got everything figured out. We're able to do our MRIs. Our MRI clinic is where a cancer center has their own protocols. Most people who come in aren't concerned. In terms of the concern about them wearing masks while they're receiving TMS, we haven't had any problems. We do ask people if they're comfortable, and we offer to pause it and things. So it hasn't, I can still see if there's like a flinch because it's too strong. It's not necessarily masked by the mask. So we are doing pretty good. We opened up our hours. Usually we operate 10 to 7. We now are trying to do 8 until 8 so that we can have more time in between appointments, but still see a reasonable amount of people, because sometimes our people come twice a day, so we have to accommodate schedules anyways. So we haven't, like right now, we just put out an ad and our phone has been ringing off the hook all day. So we've got a lot of interest and really not a ton of concern in terms of people asking what we're doing. They just come in, we take their temperature, and they just do pretty much what we tell them. They're not worried as much.
It's great. Thank you guys so much for sharing.
I'll say regarding particle flow dynamics, like coming out of the mask, we considered, a simple solution of a fan blowing downwards, but then, and then multiple HEPA filter devices along the bottom of the floor to prevent the spread from coming up is a few $100 solution, but we're not a particle flow dynamics lab. But so some of it was common sense, like finding a flow that is not too strong to blow it everywhere, but just coming up from the subject. I mean, with a vertically mounted fan and a couple of $200 HEPA filter vents on either side of the chair. So maybe consider that.
Does anyone else have anything? Xingbao, did you have something to add?
Hey, Colin, how are you?
Good. How are you?
Good. Yeah, nice to see you here. And probably this is Giovanni from Medical University of South Carolina. We typically, because the addiction, Colin maybe up to but here we never stop for the depression the trial. It's even this depression trial we use the MRI and also use EEG. And I see here we have like a standard protocol. If you want to continue your study, that is kind of involve the patient. We need to apply from university through the red cap. Then the university approve. Then for the depression patient, every day we do EEG, TMS, synchronized TMS, and MRI is it's limited to people. We normally because the MRI also include the EEG. That is like at least 3%. And for the daily TMS treatment, we have the red cap, have the RA every day to first through the phone screen make sure that patients don't have any fever and some syndrome. And when they arrive here, they also need to feel about to take like 2 or 3 minutes on the red cap to make sure it's patient is no syndrome. Yeah, that is all the procedure. And when the treatment, we just use the like glove and regulate the mask. It's no like yeah, in 95 no surgical mask. So just use the regular mask. And after each treatment, we just like clean the chair or TMS coil. Everything is just the edit like procedure to take 5 minutes to clean up all the treatment. So yeah, treatment. And I think we run it like 2. For the depression, we here the MSc we run the 2 study continue, and because you always think about this is the incentive the treatment the work for the patient. Yeah, that is the update here. Yeah, thank you for this chance to share. Yeah.
Thanks, Xingba. I'm glad to know things are rolling and enrolling at in South Carolina as well. It sounds very similar to the procedures that we have in place here. Ahmed, did you want to say a few closing words?
No, it was really exciting to have people from different labs, and I think that's basically a discussion that should be continued based on the updates that we are receiving. Right now, we have a feedback that, okay, there might be some changes in the bold signal because of the using the mask inside the scanner. And I'm not sure, at least personally, when I have kind of masks, when I'm in the kind of meetings and I'm discussing with people, I easily feel that I'm getting out of breath sometimes when I already have the mask. And I'm running marathons, so I have a really good kind of cardiovascular system. My heart rate is something like 62. So, but even with that cardiovascular system, when I have mass and I'm discussing hot topics with the students, sometimes I just feel I do not have enough oxygen. So that's the feeling that at least personally I feel that I have. And you know that there are some procedures that if you hold your breath inside the scanner, and it would just increase a little bit of your kind of pulse oximetry inside the scanner, it would change the bold signal significantly because there might be some challenges for that. So how those things are going to be affecting the outcomes that we have, I mean, these are the empirical questions that we need to explore together. And it seems that as we are all around the world are reopening labs, we need to keep on discussing together about the questions and the challenges that we have. So my suggestion is just being in touch with the INTAM kind of email address and the Twitter channel that we have. So please share your experiences, your questions, your concerns, your innovative solutions that you are right now using in different labs. And we definitely will be happy to share those experiences with emails and with the Twitter channel that we have. So please kind of sign up for the kind of INTAM Twitter channel. And that would be something that I mean, as a community, that's exactly the reason that we are here together. So we are here together to see what are the best solutions that we can make together, and hopefully we can move forward. And we might need to have another kind of the updates on the guideline that we had published in April. So we probably need to have kind of some revisions or some updates in the next month. So we'll keep kind of in touch together, and hopefully we'll make some of those educational materials and guidelines for the community in the next coming months as well. Great. So great. And thank you very much, everyone, for being with us. Thank you, Colleen, for kind of informing, helping me how to schedule the meeting. So we have been always kind of discussing about what would be the best combination of different talks. And thank you very much for being there. Whenever I have kind of a question or something to kind of discuss. And thank you very much, Dr. Walton, for joining us today, for presenting a really nice kind of ideas for kind of the next granting opportunities. And thank you, everyone, for being with us. I hope that it would be something that we will keep on doing. So hopefully we will be back together in the next 3 months for another webinar. And hopefully in the next 3 months, things would be in a better situation right now. Good. So thank you very much and have a nice day. Take care. Bye-bye.